Lecture on Cancer Immunology. Today's lecture is going to be a little bit different than some of the others because there are only a few figures from your textbook, and a lot of the figures are going to come from papers or some other textbooks. So today we're going to be talking about, first, how does cancer actually get promoted by the immune system? So in fact, some portions of our immune system can actually promote the growth of cancer cells. But then we're going to spend the rest of the lecture on how the immune system can actually fight cancer, how it does it naturally, how it can recognize tumor cells, and then how can we modulate the immune system to make it work better to fight those cancers that it's obviously failed at eliminating. So, chronic inflammation in cancer. It's known that inflammation can increase your risk of cancer. And tumor necrosis factor alpha, TNF-alpha, that you learned about in the cytokine lecture, is a major player in cancer. But based on its name, you might be able to figure out that it was actually discovered because it can kill tumor cells. Subsequently, however, it's been found that TNF-alpha actually can play a role in promoting the growth of cancer. And so we'll talk about how it can do both of those things. Also, chronic infections are a major cause of inflammation and also a major cause of cancer. So I'm going to go through a few of the major chronic infections that contribute to liver cancer, stomach cancer, and cervical cancer. But let's start with TNF. So originally, as I said, it was discovered because it could kill tumor cells. And this graph shows the different ways that TNF can help to promote killing of cancerous cells. It can do things that actually aren't really mediated by the immune system, like effects on blood vessels that cause necrosis of tumor cells or lack of oxygen getting to tumor cells, which they need for growth. It can cause mutations that cause direct lysis. It can activate some immune cells, cytotoxic T lymphocytes, which you might expect, and then also these other cells that aren't very well understood, lymphocyte-activated killing cells. They kill tumors, but they're not actually cytotoxic T cells. We won't really spend too much time talking about those today. And then also TNF might be able to synergize with some other anti-cancer therapies. So although this was how TNF was discovered, it turns out that it's probably not the major role TNF plays in cancer. So this figure shows how TNF can promote the growth of cancer. So it can actually play a role in carcinogenesis, actually causing cancerous cells to form. It can also play a role in metastases, allowing cancers to spread. And there are some polymorphisms, some mutations within the genome of humans that are known to be associated with this. The biggest difference between TNF in this graph and in the previous picture is that this is TNF at low levels. When TNF is really high, it can kill tumor cells. But at low levels, it actually promotes cancer growth. And it turns out that it's more common to have low levels of TNF than high levels, except in the case of sometimes acute infections, which those don't really, of course, promote cancer. So with low levels of TNF around in your body, that unfortunately can help tumor cells to grow. Here's a figure from a paper from about 20 years ago. It was a skin cancer model. So basically, they induce cancer, skin cancer, in the animals, and they did it in wild-type mice and also mice that are deficient in TNF-alpha, so they cannot make any TNF-alpha. So which mouse do you think is which? Which one is the wild-type and which one is the TNF-deficient mouse? So it turns out the top line in each graph, and these are all just modifications of the same system, is the wild-type mouse. So when you induce the cancer and you have TNF in the mouse will develop the skin cancer. But if you try to induce the cancer and the mice can't make any TNF-alpha, they actually won't get skin cancer. So this again shows the role that TNF actually may be more important in promoting cancer than actually killing tumor cells, as its name would suggest. Okay, let's move to infections. So the first infection we'll talk about is HCV, or hepatitis C virus. This is a virus that infects your liver, and it becomes a chronic infection in many people. So when the virus infects the cells, they're going to secrete chemokines, which you've learned about in previous lectures, and that's going to recruit innate cells and adaptive cells and cause inflammation. Now, if this immune response is productive, the infection will go away and get resolved, but that only happens about 20% of the time. What happens in most people is that they go on to have a chronic infection. This immune system doesn't eliminate the infection. So the immune system keeps trying, and more cytokines are secreted, more and more inflammation occurs, but not really a productive antiviral response. And this can eventually lead to the growth of liver cancer. Similarly, Helicobacter, a bacteria that infects the stomach, can also cause cancer. It can cause both stomach cancer and also lymphoma, but it's the same process. The immune system is activated to try to fight off the bacteria, but it doesn't tend to do it very well. So you get chronic inflammation in the stomach. You can eventually get an ulcer from that as well, but you can get stomach cancer, where the lining of the stomach, those cells actually become cancerous. In addition, you can get a B-cell lymphoma called a malt lymphoma, and that's from that adaptive B-cell going to fight the infection, not doing a very good job of that, and being chronically stimulated, so it itself becomes cancerous. So you can actually get two different kinds of cancer caused by inflammation from a Helicobacter infection. This is just another picture showing how H. pylori can dysregulate the immune system. There's a lot going on here. You don't have to look at everything, but notice over here that the infection actually inhibits helper T cells. That starts to explain why the adaptive immune system can't eliminate the infection. And it also inhibits nitric oxide production by macrophages, which should help to kill the bacteria. So what you're left with then is a lot of our friend here, TNF-alpha, causing inflammation. So this just tells you a little bit about how H. pylori can dysregulate the immune system, cause chronic infection, leading to a lot of, or not a lot, but sort of constant TNF-alpha secretion, and that can eventually lead to cancer. Another important infection that can cause cancer is HPV, or human papillomavirus. It's the same process. Again, you activate adaptive immune cells. If they do a good job of eliminating the infection, you clear the infection and that's it. But if you end up with a chronic infection, then you can end up with cancer. So, what happens with the immune response during an HPV infection? If you look over on the left-hand panel, you'll see in the blue line is the activation of the innate immune response. And that goes up very quickly, as it should, and then comes back down. And then in the dotted line is the viral load, which goes away. You can see cytokines in green get activated and then go away. And you can also see that ultimately you get B and T cells activated. That's the type of immune response you want that can help to eliminate the infection. But in the center panel is what happens when the infection becomes chronic. And the problem that maybe you can see is that there's never a good innate immune response. Now, if you think back to all your previous lectures, you'll know that it's the innate immune response that stimulates the adaptive immune response. And if you don't get a good innate response, then you'll never get a good adaptive response. And that's what happens in this case. And over here is just showing that vaccination can help to mimic what happens in a productive response, if you think back to the vaccine lecture a few lectures ago. Okay, so overall, so far we've kind of talked about how the immune system can contribute to the growth of cancer. And then ultimately, that's because of inflammation. So what this picture shows you is that if you start to have damaged cells and you activate an innate response, but you don't get a good activation of your adaptive response, this innate response and inflammation will actually promote tumor growth. But if you can get adaptive immune cells activated to fight off the cancerous cells, then they can actually prevent the growth of cancerous cells. So now we're going to switch gears and talk about how the adaptive immune system can recognize and kill tumor cells. I want to go back, though, to a graph I showed you in the very first lecture. And it talks about how the immune system decides whether to respond or not to different things. And if you remember, the immune system wants to always be turned on. So its default is to respond all the time, which of course that's not always a good thing. And that's what's shown in this table. So with an infectious agent, the normal response is you get immunity. A deficient response is you get infection. An innocuous substance you actually don't want an immune response to, and if you have one, you'll have an allergy. A grafted organ, our immune systems weren't really designed for that, but of course that's foreign, and if you allow the immune system to respond, you'd reject the organ. A self organ, though, you also don't want an immune response to, and if you get one, that's autoimmunity. Now with tumors, the normal response is going to be immunity, and a deficient response is going to be cancer. So we'll talk about how you can get tumor immunity and what goes wrong that eventually leads to the development of cancer. So what's our evidence that the immune system can help to control tumors? Because all we've talked about so far is how it can promote cancer growth. But how can it stop it? It's known that there are always immune cells found within tumors. So for decades, we've known that immune cells are attracted to a tumor, suggesting that they might be fighting the tumor. Early experiments with transplanting in a tumor and getting rejection and then a memory response found that you can develop a memory response that can help you fight off a tumor. I'll show you that on the next slide. And also, in both immunodeficient people and animals, there is an increased incidence of cancer, suggesting that if your immune system is not working properly, you will get more cancer. So this is an old experiment. But what they did was they took laboratory mice, they injected in tumor cells. The mouse grew a tumor. They did surgery and removed the tumor. The mouse was cured. They came back and they tried to inject tumor cells again, and those tumors didn't grow. The mouse had a memory response to the tumor, and it could fight it off the second time around. It's just like a memory response for an infection. In the second line, you don't actually have to give the mouse a tumor. You can just show the mouse tumor cells. Again, very analogous to the vaccines we talked about for infectious diseases, where you could heat kill an organism and give it as an injection, and that person can make an immune response and a memory response that will protect them. And that's what's shown here. The bottom line just shows that it's specific for a certain type of tumor. And that's what you would expect with a memory response. It's an adaptive response and very antigen specific. So I actually want to take you through a little bit of the history and some important landmarks that occurred in sort of tumor immunology. And we'll go back to 1891, where William Cooley found that people who had cancer who happened to get a bacterial infection sometimes had their cancer cured, suggesting that stimulation of the immune system, even nonspecific to the tumor, might help the body eliminate tumor cells. About 20 years later, sort of what we consider the father of immunology, Paul Ehrlich, suggested that yes, immune cells do patrol and are found in tumors, and subsequent research has definitely confirmed that. The mouse experiment I just showed you was done in 1953, so quite a while ago. In 1957, interferon as a cytokine was discovered, and it was used as a treatment. Now, it did work somewhat. The problem is interferon was so strong, it actually stimulated the immune system too much, and that was very detrimental for patients. But the idea that you could turn on T cells and help fight the tumor was proved with these studies. And again, a different type of infection experiment, going back to our TB vaccine, if you give someone a TB vaccine and you stimulate their immune system, that can help fight off a tumor, and this was shown in mice. 1973, dendritic cells were discovered by Ralph Steinman. He eventually won the Nobel Prize for this. He was actually at Yale when he did this work. And dendritic cells, of course, are important for lots of things in immunology, as you've learned throughout this lecture series. But as you'll see, they'll be very important for trying to stimulate anti-tumor T cell responses. It was 1983 when the T cell receptor was discovered, and that can help, once you understand the T cell receptor and MHC, which you've learned about, you can start to understand anti-tumor responses. 86, we had the first antibody drug, and later on in the lecture, I'll tell you how antibodies can actually be used more as drugs to try to treat certain diseases. And basically it was rituximab was used in 1997 to cure some lymphomas. And in the last 10 years, we actually have our first cancer vaccines, but I'll talk more about those coming up. So, using the immune response to attack tumors. As we know, our immune systems are capable of developing an adaptive memory response to tumors. So we have something to emulate and mimic to try to improve getting more immune responses to more tumors so fewer people have cancer. Now, if this worked all the time, no one would get cancer. So obviously there are mechanisms that the tumors can use to evade immune responses. We can identify antigens to which our immune system can respond. We can use monoclonal antibodies. These antibodies are going to be used a little bit differently than we think about antibodies in a true immune response to an infection, and we'll go over that. And then there are ways to try to enhance an underdeveloped immune response to a tumor or to relieve suppression of a developed immune response. So, how can tumor antigens be recognized by our immune system? Because remember, tumor cells are cells. However, what makes them tumor cells is that they acquire mutations. And those mutations can help to make some of the antigens in those tumors look foreign to our immune system. So that's what's shown here. You can have a mutated self-protein, mutated oncogene or tumor suppressor gene, which are proteins, of course, and you can get different peptides presented on MHC, and those will be foreign, and you can activate a T cell response. And of course, if you think about some of those infections I talked about that induce cancer, well, they of course have foreign antigens with them that hopefully could stimulate the immune system. I want to take you back to one of your earlier lectures, and I want you to really think about the T cell receptor and MHC, how they interact, and how a peptide sits in an MHC molecule and how it interacts with a T cell receptor. Because this is going to be important when we talk about all the different mutations that could occur that could allow tumor antigens to be recognized. So, in a normal cell, it's always presenting its own self-antigens on MHC class 1, but T cells learned in the thymus not to respond to those, so there's no response. You can have point mutations that occur in antigens and that occur in order to make cells cancerous. And you could actually have a new peptide presented on MHC to the T cell receptor. You can also have a peptide that never got presented before, but now with mutations has gained the ability to bind to MHC and be presented for the first time. So if I go back to this slide, the difference in those two scenarios is in one, the point mutation occurs here on what the T cell receptor sees. So it sees it as a new peptide. In the second scenario, you had mutations that now create anchored residues, allowing a peptide for the very first time to sit inside the MHC molecule. Both of those would look like foreign antigens, and T cells should be able to respond. There are a few other ways cancer cells can be recognized. One is that cancer cells often turn on stem cell or germ cell genes that were only ever expressed during development. And once T cells started to be educated in the thymus, those germline genes were never turned on, so those proteins were never made. So our immune system has never seen them and never learned that they were self. So again, they can look foreign, and you can have T cells that react to those antigens. And sometimes just having too much of a self-antigen can be enough to stimulate a T cell. If all of the MHCs have the same peptide, even though it's self, sometimes that's enough and you can get a T cell response. Well, with all of these ways that our immune system and our T cells in particular can recognize tumor cells, why do we have so much cancer? Because every time someone's diagnosed with cancer, by definition, the immune system has failed to eliminate the tumor. Now, what we unfortunately have no way of measuring is all the successes of our immune system. We all, right now, today, each one of us has cancerous cells in our body that our immune system is eliminating. But we'll never know about those because they eliminate them before they ever become a problem for us. Just like eliminating an infection from a memory response, you never get sick, you never know you were exposed again, your immune system eliminates that threat, and you have no idea that it was even happening. So, how can tumors evade immune responses? Well, you can lose the antigen to which the T cell responded, and then there's nothing for the T cell to see if there's nothing presented on MHC. Sometimes it's MHC itself that's lost. Mutations can occur in MHC or the pathway to help it get presented. And sometimes, and we'll get to this at the end of the lecture, you can have your tumors actually induce an immunosuppressive state so that even if you have T cells that want to kill the tumor, they're sort of turned off at the last minute. So if we think about tumor cells and the immune system, we mostly have T cells and K cells and then some of your innate immune cells. And Actually, both tumor cells and immune cells can secrete cytokines that have an effect on one another. If you get a productive immune response, you get good anti-tumor immunity. You'll get things like IL-12 and also interferon gamma. You'll activate NK cells. You'll activate cytotoxic T lymphocytes, and you'll eliminate or suppress the growth of the tumor. If instead you get inflammation and you get different cytokines, like our old friend TNF, you actually can have tumor growth. Now, cytokines, just like TNF, they're not always all good or all bad, and IL-10 is another example of that. IL-10, as you learned, is an immunosuppressive cytokine. In the case of chronic inflammation, that's a good thing, and you can suppress the inflammation and therefore prevent tumor growth. But if a tumor secretes IL-10, then it can suppress the productive anti-tumor immunity, and that would be bad. So how does the immune system lose control? Because for most tumors, the part we don't ever know about is that for some amount of time, our immune systems were controlling the growth of that cancerous cell. And we only know when they lose control. So this just shows how you get cancerous cells. And here again is what happens if you have a productive anti-tumor immunity. You get your CD8 cytotoxic T cells, NK T cells, NK cells, and then some innate cells to help stimulate those adaptive cells. You get good cytokines made, and the tumor is controlled. So again, all of us might have some tumor cells like this right now in our body. They exist, but the immune system is controlling them. But what tumor cells do is to continually mutate and evolve. And so what at first in this sort of orange color here was highly immunogenic, mutates so it's now poorly immunogenic, and our immune cells don't really see that tumor cell anymore. And then eventually they change enough that not only does the immune system not see those tumor cells very well, but even if they do, the tumor has induced suppressive immune responses like Treg cells to suppress any immune response that is generated. And this slide just shows again the different possibilities for how the immune system can lose control of tumors. Again, the first two are that there really isn't anything for the immune system to respond to. You just can't make a good anti-tumor response. Even if you had a good response, the tumor mutates and now that antigen is gone. Infections do that as well. Viruses are very good at mutating away from adaptive T-cell responses. This panel shows you again how you can have a cytotoxic T-cell. It's there. It wants to kill that tumor cell. It's interacting with that tumor cell, but it's being suppressed. It's not allowed to complete its job. And you can also actually have a physical barrier. This is not immune-mediated, but this physical barrier created by tumor cells can actually prevent immune cells from getting to tumors.